Hi, my name is Maksim Mamonkim from Baylor College of Medicine, and I am quite optimistic that we are getting there in terms of CAR T cells for T cell ALL and T cell lymphoma. Obviously, it is difficult to match the efficacy and safety that we are observing in B cell malignancies in B-ALL and B cell lymphoma, and extending this approach to T cells. Requires solving additional problems such as limiting fratricide of CAR T cells and reducing ablation of endogenous circulating T cells. But we, as a community, has made pretty good progress lately, developing and evaluating CAR T cell therapies in T cell ALL. We started to get very encouraging results. Our colleagues in China have evaluated several approaches in CD7 CAR Ts using various methods of fratricide prevention, such as gene editing or trapping the protein inside a T cell. And they're able to manufacture these products either from the autologous material or from prospective donors or from completely off-the-shelf and third-party products. They reported very encouraging results in patients with refractory and relapsed T cell ALL and lymphoblastic lymphoma. Reporting 75, 90, 95% responses in this very difficult to treat disease. Also, there's a very encouraging report recently from the UK using CRISPR-based editing off the shelf CD7 CAR T cells, we're also producing very good, very promising responses. So I believe that we're heading in the right direction. My group has developed unedited CD7 CAR Ts that don't require any additional steps to prevent fratricide, where we expand them with pharmacologic inhibitors. And so it's a very straightforward process. And we also have an ongoing trial at Baylor here in Houston in patients with refractory relapsed T cell malignancies, including T-ALL, which is open to both adult and pediatric patients. And we are seeing similar responses as our colleagues across the pond. So we are optimistic that these options will be soon available to patients with T cell ALL and lymphoma. There are still several problems that we need to solve, primarily those that concern T cell aplasia. And luckily it is limited because these patients do still retain the T and NK cells that essentially lose CD7 expression. That's how they avoid being eliminated by the T cells. But these patients still are susceptible to bacterial and viral infections, which are sometimes fatal, unfortunately. So we need to be mindful of that and design our therapies to have limited duration and allow the T cell compartment to restore or proceed to transplant as soon as possible. Antigen escape has also been noted in several patients, pretty much mirroring what we see with CD19 and B-ALL. So other approaches targeting additional antigens definitely needed. And speaking about additional antigens, we're also exploring CD5 as a target in T-ALL and also in T-cell lymphomas. It is very highly expressed in mature neoplasms in peripheral T-cell lymphoma and cutaneous disease. But in T-ALL is also very highly expressed in most subtypes of ALL. And we have a clinical trial ongoing at Baylor using autologous CD5 CAR T cells or those derived from a prior stem cell donor. We will manufacture them and give to patients with refractory relapse disease. And we are seeing very encouraging response, particularly after we have optimized the manufacturing and incorporated some ways to remove or reduce tonic CAR signaling in these T cells. And we are seeing 75% response rates in patients with very bulky disease very frequently. And so the products produce very good safety profile, pretty much on par with what we see with CD19 products in B-ALL. They're not very inflammatory, and they persist enough to eliminate the disease, but not forever. And so this would be a great alternative option for T cell ALL. And so we're excited also to move this further to phase 2 trials very soon. So This was a sort of very expanded version of the answer, but the short answer is yes. I think we're getting there, where both CD5 and CD7 targeted approaches will become more widespread and available to patients with TLL.